In this video, we will explore the complex process of large intestine development. We will discuss regional boundaries, midget loop morphogenesis, rotation and fixation, guidance by structures like the dorsal mesentery and caudal mesenteric artery, cellular differentiation, patterning, and epithelial mesenchymal interactions. Join us on this journey to uncover the secrets of large intestine development. In summary, the formation of the foregut midget boundary is a crucial event in the development of the large intestine. It involves complex molecular signaling pathways and plays a vital role in shaping the structure and function of the organ. Summary: the midget loop undergoes a counterclockwise rotation during development, which positions it correctly for the development of the large intestine. Malrotations can lead to intestinal malformations. The rotation and fixation of the large intestine during embryonic development ensure its proper positioning in the abdomen. Failure in these processes can cause complications like volvulus and intestinal obstruction. The development of the large intestine relies on the dorsal mesentery for guidance and organization. It also aids in the formation of blood vessels that supply the large intestine. Issues with the dorsal mesentery can affect the viability and function of the large intestine. The caudal mesenteric artery, CMA, is essential for the development and blood supply of the large intestine. Malformations or branching issues can lead to blood flow problems and developmental abnormalities. Exploring CMA development could lead to treatments for large intestine vascular issues. During embryonic development, the hindgut differentiates into distinct regions that give rise to various segments of the gastrointestinal tract, including the large intestine. This process involves the establishment of molecular signaling gradients and the subsequent activation of specific transcription factors. The hindgut is initially specified by the expression of key developmental genes, including CDX2 and HNF4A. As development progresses, the hindgut becomes regionalized, with different regions acquiring specific characteristics necessary for the formation of various segments of the gastrointestinal tract, such as the large intestine. This regionalization is governed by the precise activation of transcription factors and specific signaling pathways, such as the WNT/beta-catenin pathway and the hedgehog pathway. The formation and patterning of the large intestine along its anterior-posterior axis are crucial for its proper function. This process involves the precise regulation of signaling pathways, the expression of specific genes, and the establishment of morphogen gradients. The patterning of the large intestine is influenced by various signaling pathways, including WNT, BMP, and FGF signaling. These pathways regulate the behavior and fate of cells within the large intestine, ensuring the correct positioning and differentiation of specialized cell types along the anterior-posterior axis. Perturbations in these signaling pathways can disrupt the normal patterning of the large intestine and contribute to developmental disorders. Embryonic development of the large intestine involves intricate interactions between the epithelium and underlying mesenchyme. These interactions are crucial for providing the necessary cues for the correct morphogenesis, differentiation, and patterning of the large intestine. The epithelium and mesenchyme communicate through various signaling molecules, growth factors, and extracellular matrix components. The reciprocal interactions between these two tissue types orchestrate the proliferation, differentiation, and migration of cells within the large intestine. Disruptions in these interactions can lead to abnormalities in large intestine development, such as altered cell fate specification or impaired tissue organization. WNT signaling is essential for large intestine development. The WNT ligands and receptors are expressed in a gradient, controlling cell fate and proliferation. Dysregulation can lead to developmental abnormalities. The large intestine epithelium is composed of various specialized cell types, including absorptive enterocytes, goblet cells, enteroendocrine cells, and tuft cells. The formation of these different cell types occurs through a tightly regulated process of sequential differentiation. Progenitor cells within the epithelium undergo proliferation and subsequently differentiate into the different cell types of the large intestine. This process is orchestrated by the precise regulation of signaling pathways, such as Notch signaling, and the sequential expression of specific transcription factors that drive cell fate determination. The proper differentiation of the large intestine epithelium is essential for its functional integrity and the maintenance of its absorptive and secretory capacities. Developmental disorders, like Hirschsprung's disease, affect the large intestine. Genetic and environmental factors play a role in its development, leading to the absence of ganglion cells and colonic motility issues. Symptoms include constipation, obstruction, and distension. Early diagnosis and surgery are important for proper treatment. The large intestine has structures called intestinal villi and crypts that increase surface area and support nutrient absorption and stem cell function. Molecular events mediated by signaling pathways and specific factors control the formation and maintenance of these structures. In conclusion, the development of the large intestine is a complex process involving precise morphogenesis, regionalization, and cellular differentiation. Molecular signaling pathways play crucial roles in regulating cell fate and proliferation. Understanding these mechanisms is essential for diagnosing and managing developmental disorders affecting the large intestine. As future medical professionals, gaining knowledge about embryological processes is crucial for comprehending clinical presentations and providing effective treatment.